So to continue our discussion on research and science, we would now like be conducting a live question and answer session with the members of the Alliance Scientific Advisory Council. We are very proud that there are 5 members joining us today. For me, it's been a pleasure of being the liaison of the board of directors for the Advisory Council, and I couldn't be prouder. There are 2 other very important councils that I like to put out. First of all is, of course, the Patient Care Advisory Council with patient and carers in there. And the second one is the Innovating and Technology Council. Back to the Scientific Advisory Council, we have 10 members in there. And today we have on the panel David Taylor, who is the chair from Canada. Then we have Gethin Thomas, which we already heard from Australia, Nick Cole from England, Caroline Ingre from Sweden, and Kuldip from the United States of America. Also in the Scientific Advisory Council are Janine Heckman from South Africa, Adriano Chio from Italy, Luis Barbaito from Uruguay, Kyun Lu from China, and Piera Passinelli from the United States. So we really have a worldwide view, or they really have a worldwide view on what is going on around the world. So that is, of course, a great situation. This is a great opportunity for all of you who are joining the meeting to pick their brains and ask some of your burning questions. To ask questions, you can enter them in again into the Zoom screen or click on the participants and raise your hand, and we will try to get all the questions that will be put out there. We hope you can participate and enjoy and get informed. And let me be unpolite and start off with my first question, which is I'm already here now in this position from 2014. That was the first time for me was the meeting in Brussels. And we're now, of course, into sort of virtual Montreal. So I'd like to ask to the chairman, which is David, what is the speed that we're in the last few years compared to the things that has been done before Brussels? David.
Well, thank you, Jorjan. Yeah, I mean, so the thing is, every year we get more and more momentum. And if we're talking about the past 5 to 7 years or so, there has been a tremendous advancement in our understanding of ALS. We still have a long way to go. It's an incredibly complex disease, but one of the things we've learned in the past decade or so is that what's happening in ALS is actually our understanding of ALS is happening at the same time as understanding a whole new set of biology that we didn't understand as science or research before that. And I'll pass this off to my colleagues as well on the panel, but the other piece is tremendous advancement in terms of clinical trial design and the number of engaged companies and targets that will be coming forward for ALS clinical trials. So if we look now, there are so many companies, so many small startups, so many targets that are being advanced for clinical trials in ALS, and starting to see ones that are coming out of that work that we've understood in the last 5 to 7 years. So the targets are getting more exciting. We're getting more interested in seeing things that will be more specific to ALS. And in fact, we have trials for the first time now that are actually targeted on molecules or substances that are derived from specific ALS research. So that's exciting in my mind. I don't see my panelists, Jorjan, but I think they're here. So if any of them, there they are. Hi, everyone. Anyone would like to add to that? Good to see you all.
Okay, let's start with the women, Caroline, because I see that there we have 4 men and 1 woman. So up to you. What is your view on the last years on research?
I really agree with David's comments, and I'm really excited. I think there has been a nice work in collaborating all over the country. I mean, in Europe, in Asia, in America, in Canada, everywhere. There's this willingness to interact and to share your knowledge and to work together. And I think that's one of the most important parts or comments thinking about the last couple of years. And I also think that it's extremely interesting that we are trying to reach some kind of precision medicine in ALS as well, trying to target specific traits or specific differences among the patients, and then treat patients with specific phenotypes, finding them all over the world and coming together in clinical trials. I think that's a very interesting way forward.
Thank you. Nicholas, are you?
Yeah, hi, thank you. Thanks for this opportunity. Yeah, I think, I mean, what's changed in the past few years, obviously we know a lot more about the biology of the disease, which is really, really important. There's been lots of additional trials that are ongoing and very exciting ones. We've had some results coming out and there are trials progress. I think one of the real bonuses is now that some of this work is a little more patient-centric. So there's optimism in the future that exclusion criteria will be slightly reduced in some of these trials that come forward, so more people will be able to get involved. And I think we've kind of seen a real positive in the way that, as said at the beginning, that companies as well as patients and also the researchers are all kind of working together and collaborating. So yeah, there's been some really good positive changes and steps in the right direction. Hopefully that'll keep continuing as we go forward.
David, you are the moderator from this session, so it's up to you.
Okay. All right. Well, certainly if there are any questions, please add them to the chat box and we'd be happy to answer those. When will we stop collecting genes connected to ALS and start using them for treatment? Okay, thank you, Gethin, for that question. I would like to say that I think we are using them for treatment and things are moving pretty well in that direction. I think as you know, since Kuldip has not spoken yet, is that possible to pass you off to that one, or maybe Gethin? I don't think we have Kuldip right now. Gethin, how about you?
Yep. Yeah, well, obviously we've got tofersen out there at the moment, which is targeting SOD1, and as far as I'm aware, I think Biogen have gone into phase 1 trials with drugs targeting FUS and C9orf as well. So I think there is already some translation directly from that genetic knowledge, but I think as we heard in the previous discussion, it's going to be precision medicine, and if we can keep identifying genes as well as identifying specific genes to target, you're also identifying pathways, I think, and I suspect that once we can prove, hopefully we can prove that say SOD1 pathway is a good target, then that might expand beyond the familial cases and into the sporadics as well. It might even provide drug targets in that area. So I think it's important to keep finding more genes, but I think we are in the process of establishing that the gene targeting is a very viable approach.
Anyone else like to add?
Yeah, I'm happy to come in on that. I mean, hopefully we won't stop collecting genetic information until there is no more ALS MND. And, you know, it's not just about particular genes. Gethin mentioned there, tofersen, which will hopefully be successful in a proportion of people with MND, but there are also now these genetic targets which are looking towards all ALS MND, sporadic cases included. So there's, for example, the Ionis supported by Biogen trial of ataxin-2, which is hopefully going to remove the TDP-43 protein. So yeah, hopefully we'll continue gaining more information about what causes the disease. Obviously, it's very complicated, and yeah, so hopefully that will continue until we know everything we need to know to find an effective treatment.
Anyone else want to add?
Yeah, David, can I comment?
Of course, of course.
So I think it's a fair question, not to just gather information, but also to use it for treatments, since that's what we're lacking, right? So, but there's a lot of things to learn from the gene collection, and there's also not dominant gene, but genes that maybe can be influenced by connecting them to environment. So there's a lot of steps left to do with gene research that we haven't done yet. And we need to figure out why some patients actually express the disease, or some people express the disease if they carry the gene and some don't. So we need to understand the gene, the penetrance patterns, and we need to understand if carrying specific genes, even if you attract the disease or get the disease, it can influence your the way you have the disease, or the way the disease will progress. And that's a very interesting information trial-wise. So I think that we need to keep on collecting them. But of course, as the other speakers have said, it's really, really interesting that we are targeting specific genes now, and trying to filter out patients that will actually respond to specific treatments. I think that's the way forward.
And I would echo as well that the Ataxin-2 is an interesting landmark for ALS, because this is a time when we're now taking something that we've learned genetically and we're actually targeting that for sporadic ALS, which is something 5 years ago we would never have really thought of. So I think that that's pretty exciting. And the other thing that I would add to that as well is, every time we discover a new gene, what's exciting about that is, if we haven't worked with that before in ALS in the laboratory, what we can do is we develop models of the disease, sometimes in animals, sometimes in cells, and then we look at what happens and what that gene, what the change in that gene does in that animal or in that cell will ultimately start to form all these new connections for us with all the other ones that we've looked at, and say that might be a new therapeutic target that we should go in and try to slow down ALS. So every time we discover one of those, I think it's a big, exciting piece, because it's going to be another piece of the puzzle for us to both think about targeting clinically, which is super important, but also finding new clinical targets that may not be that gene directly. So it's I think, as the colleagues have said, discovering new ones is still very important, but definitely activating them is key. I see that we have another question. And I think it was Dirk, had asked about whether we will ever have a global regulatory agency. So for ALS, obviously this would be an amazing thing to have, something that could consider and approve or not approve clinical trial results for a global community or a wider community. And I see that our colleague Kuldip is there, so maybe I'll pass this one off to you. The question is whether or not there may ever be a global worldwide regulatory agency that provides market approval for all continents, or whether they should stay separate. The FDA, EMA, etc.
Yeah, can you hear me now, David?
Yep.
Yes, we can hear you.
Okay, great. Sorry about that. I was having some technical issues earlier. It's a great question, and I think from a convenience perspective and generalization perspective across the world, that's a good idea. Unfortunately, I don't think that's a possibility. I think every country has their own way of determining whether a drug can be approved. They have different criteria based on the populations they have. So it's a great idea. I just don't think that operationally it's possible. But you never know what happens in the future. David?
Well, any other panelists have something to say, please do speak up. I would say that one of the things that Canada has been able to form with a few other countries with regard to cancer is something called Project Orbis, which is a harmonization where FDA, EMA are going to be the two top hits that any company's probably going to go towards for a market. And So other markets, Gethin, I'm looking at you, and others that are maybe not connected to the larger ones, are going to be wondering, you know, when do we get a therapy? How much longer would it be? And I think these kind of potential partnerships where other regulatory agencies can look to the FDA, look to EMA, find some parallels might be possible in the future, but I think we need to also be able to inform our regulatory agencies about ALS trials, what they mean, what are the outcome measures, what constitutes a good trial result versus another. And the more that we can inform them and the more that they can find parallels with or partnerships with FDA and EMA, I think that there's possibilities for this down the road. And it speaks to the amount of advocacy and education that we need to do individually in our own countries to try to align with that, because we inevitably the FDA and EMA will be the places that most companies or new drugs will go to first. So it'll be on the rest of our communities, I think, to find alignment with that. That's my suggestion anyway. Anyone else care to speak to that, or would you like to move to the other question?
David, have you seen the question or remark from Michael Robinson, Project Orbis? Can you say something about Project Orbis?
Yeah, so that's what I was sort of referring to. I think it's a wonderful concept. So Project Orbis is a collaborative effort where Canada, Switzerland, Singapore, was it Australia? I'm not sure if it's Australia. I apologize. Have aligned with the FDA for certain cancer drugs to be basically co-reviewed and potentially co-approved. Now, in cancer, there's one additive benefit in that they have some very objective outcomes for their trials. They have tumor-free survival. They have things that are very well established. ALS, we have some good outcome measures, but they're more subjective in their interpretation, which you will see by even just listening in on an SAC discussion on a latest clinical trial result, where there's nuance in how we all will interpret those results. And so I think getting to better education about the outcomes of trials in ALS, getting better biomarkers that are objective in clinical trials for ALS that can tell us whether or not something is truly working, are going to be important for those kinds of alignments to be easier, so that the opinion of the FDA or the EMA would also align with other countries and their regulatory bodies. That would be my sort of thought process on Orbis and the potential down the line. Anyone
David,
else?
there was a question from Amaji. What is the most promising research therapy modality which is nearest to clinical application by doctors for ALS patients?
So I'm guessing that's what's next in terms of excitement or things that we are excited about for clinical trial and potentially either moving out of the lab into the clinic or in the clinic now. So I will pass that off to my colleagues if anyone would like to volunteer some ideas of things that they're excited about that's either in trial or will likely be in trial soon as a target. Do you want me to pick on somebody? Nick looks like he's ready to talk.
Okay. Yeah, so it's there are many promising therapies in progress. I mean, it's hard to say which one we think is going to be the most successful, because it wouldn't be right to say that until all of these things have been properly tested and gone through all of the right processes. But certainly the fact that there are many treatments in phase 3 that are going through, and I guess they're just the same as anybody would think, you know, those would be, they've got to that point in phase 3, so they'd be the most promising, and we await to find out what the data says. Yeah, so there's a few still in progress that are fully recruited, which is amazing given the issues with the COVID-19 situation. You know, a few I could mention, but we may come on to those later. So yeah, I don't think it would be right to say exactly which ones. I mean, I can only go with what's been reported that everyone else knows about, really.
Thank you, David. I see a question from Philip Green, who is in the Patient Care Advisory Council. How close are we to having validated biomarkers in ALS so we can have more objective endpoint measures?
Amazing question, Bill.
Yeah.
Okay, so I'm going to open it up to the panel if anyone wants to talk about any particular things they're excited about, because we'll still keep that going as a potential. And at the same time, in terms of objective biomarkers, anyone want to chime in? Caroline, perhaps, or?
Yeah, please.
Yeah, that would be great.
So I think this is a really, really interesting question. And I actually think that it connects to the question about clinical trials, because if we can find biomarkers that are exclusive for some patients or subgroup of patients, we might also find treatments that are exclusive for some patients. Maybe we should think that the patients are not as similar as we have thought. Maybe ALS is not a homogeneous group, it's a heterogeneous group. So are we really aiming to find a treatment that will treat all patients in the group, or should we aim instead, as we have been talking about, to find SOD1 genes and treat the gene carriers with antisense treatment, and then maybe treat the people with higher inflammation, biomarkers for inflammation treatment, and so on. So I think this is a really big and really, really, really interesting question. There are a lot of biomarkers coming out, both in blood, cerebrospinal fluid, but also dry biomarkers like imaging, MRI of the brain, PET scans of the brain, and maybe we can connect those together with the biomarkers in the blood and cerebrospinal fluids, and then find specific patterns for subgroups of the ALS patient community, and treat them accordingly, and then follow the efficacy of the treatments. I think that's the way we should see it, and there are things coming out. I think that we just need to maybe recognize the heterogeneity of the cohort, and find different biomarkers instead of one biomarker, and different clinical trials or treatments instead of one.
Anyone else? I think it's also very, I think Gethin, perfect.
Yeah, I also think it's pretty exciting that a lot of clinical trials now are routinely measuring biomarkers. So that's going to increase the body of data that we have to ensure that we're not using inaccurate biomarkers as well, and making sure we're using only the biomarkers that can really tell us information. I think that's really important as well, that the more data we have, the better we can identify which biomarkers tell us useful information.
Gethin. Yeah, I think all the big initiatives that we have around the world where we're observing people living with ALS. So you've got SALSA in Australia, you've got Ambrosia in the UK, you have multiple ones with TriCals, Answer ALS. All of these are going to help us to get better understanding of humans and be able to look for biomarkers of progression of the disease that are going to be more objective. And as we come together, like Caroline said, in the world and find these, sharing that data and coming together, we're going to learn more about that. And I think the other thing is, we shouldn't be accepting clinical trials where we don't see some sort of biomarker of the outcome of what they're looking for. So if you're treating with a drug that's supposed to do this in the body, we need to make sure that those companies are having some sort of biomarker that says it actually did that. Because if it works or doesn't work, we need to know whether or not it actually hit the target it was supposed to, so we know whether that's a good target moving forward or not. So biomarkers are probably one of the biggest things that we can push in ALS to really get to a better clinical trials, and of course the regulatory agency piece afterwards. Were there other questions? I know there's more coming in. Jan, I'm not sure if you see any.
Yeah, and the news on Project Mine.
Sure. Does anyone want to answer that? I'm happy to answer that.
Yeah, I can take that.
Perfect. Go for it.
So for everybody, Project Mine is a unique project. It's an international project that is doing genetics ALS research across the world. So again, I think David mentioned earlier, the first step in drug development is to identify targets, something in your cells, in our bodies, that is different, that's changed. Genetics and gene sequencing allows us to look at what genes, mutations may be either increasing your risk of ALS or reducing your risk of ALS. And this is why these gene sequencing approaches like Project Mine are very important. What Project Mine is going to do is collect over 22,000 DNAs from patients, and then sequence them. I think as far as I remember, they have sequenced 50% of those that have been collected so far. And so that's where the project stands. The other piece of this is that it's great to have gene sequencing data, but what's also important is how does this data get socialized? How does this data get available to all the researchers in the world? And so, for example, the ALS Association supported a project with John Landers where we're taking this gene sequencing data sets and putting it all in one place so that then the rest of the community, research community, has access to that data and can analyze that data and look for more targets and identify more gene mutations. So that's where the project stands as of today. David, back to you.
Anyone else want to add to that? I think. Oh,
yeah, I'd just say again, it's a really great example of collaboration that's going on between researchers and people affected by MND, and it's not finished by any means, and that data is there and being continually mined, and there are new results coming out all the time, and new publications, and building on those foundations are where we get to with our start of therapies and trials. So yeah, there's news coming all the time and analysis of that data. So it's a phenomenal resource and a really great example of, as I said, people working together. David.
Yeah, and I would echo that. The other thing about Project Mine and doing what they're doing, which is called whole genome sequencing, which is very exciting, and bringing off what Gudjon asked before, is what we've looked at for most of our ALS research time so far is about 2% of all the genetics of a human. And we've learned so much from that 2%. And now what Project Mine will do in these whole genome sequences is it takes in that other 98% of our genetics that are very complicated and that we still don't fully understand, but we're going to have this resource for the next several, like many years ahead, and starting right now, obviously, to really understand all the really nuanced things that aren't the simple genetics, and how that's going to help us to understand why that person has ALS genetically and that person didn't, or what made that person susceptible and that person not susceptible. And I think that that's one of the powerful things that we're only touching the tip of the iceberg with, that Project Mine and these collaborative efforts are going to yield much better understanding of the disease in the years to come. I'm excited about that for sure.
David, a question from Karim Maestrik. Just to put out how we work with the board of directors and the scientific advisory council. So if we do have questions on potential medicines, we do we ask you as the scientific advisory council to give an opinion on potential drugs. And on the website of the Alliance, underneath the advocacy, you can see there are some briefing notes from you as the Scientific Advisory Council, which is the opinion of the 10 Scientific Advisory Council members on potential medicines. And I really like to put out to everyone just to read them. You don't have to agree upon them. It's just like it's the information that we like to share with you. So a patient would like more access to that information gathered. I can understand. So the information that we have and share from the Scientific Advisory Council are on the website underneath the underneath the advocacy tool. Recently, there has been, of course, some information about Neuron, Brainstorm, and of course, Amylyx. And could you please tell us a little bit about the process, how it works with the Scientific Advisory Council if you do get a question from the board of directors to make a briefing note?
Sure. So essentially what happens is when there are questions that come forward from the board of directors, whether that's a member of the Alliance who wants to ask a specific question through the board, and then the board comes down to the SAC, or sometimes they're so obvious, Brainstorm, Amylyx being ones that are either driven by the SAC and say, look, would the board like something like this? Or the board will ask us to do that. And then this amazing group of people from all around the world with amazing expertise on the SAC, we go through a process of usually through email, but we also do have quarterly calls where we start with a first draft and we workshop this. And this brings in all the different opinions, not just of the members on the council, but all their connections as well, to sort of really come down to something that we feel is the most appropriate answer we can give back to the board to put out to the members. And so it's a pretty robust discussion. Nobody shies away from a different opinion. And I think that's one of the great things about the balance of expertise on this panel. It's such a privilege to get to work with them all, and to learn from them all. Jan, was there anything in particular? Then we send this back to the board, and they ultimately will, as a recommended briefing note, and they will ultimately approve whether or not they decide this is what they would like to put out as their message to the members.
No, thank you for answering that question. I have a question from Alper Kaya from Turkey. Any multinational collaboration research based on genotype and phenotypes?
Okay. Does anyone want to answer that in terms of?
I can answer, or
Perfect.
I can start.
Sure.
So that's a very interesting question. I think it goes along with what I've been talking about before. There is a big collaboration. Now I'm talking about Europe because I'm placed in Europe. So when we are talking about the TRICALS, which is Trials Against ALS, a consortium of different countries working together to find a treatment. And in this consortium, we're working a lot with prediction models and precision medicine. So trying to find the clues to why the phenotypes are different. And if the phenotypes are different, do we have any specific biomarker, like a genetic marker or any other biomarkers that will stand out and make us understand that it's one specific subgroup of patients. So there is there is talk about launching a trial where in which some patients will be carrying a specific genetic mutation, not causing ALS, but that mutation means or will mean that the patients will have a specific, much better response to a specific drug. So if you're not carrying that specific gene, you will not respond to the drug. So the TRICALS consortium, they're trying to find people or patients that will respond specifically to specific drugs, and then try to find those patients so that each therapy will be specific for that patient instead of treating them all. So I think that's a very nice collaboration between very many countries to find eligible patients based on their specific criteria.
Thank you. Could you tell something about the question of Michael Robertson, David, or ask somebody from your scientific advisory council to answer that one?
Sure, I will see if I can. Okay. Are there any major initiatives resulting from collaboration of ALS organizations globally? Well, yes, absolutely there are. And I think we're probably, and this is actually stems off very nicely from what the last question had as well, which is, I think, and what Caroline said earlier, we are becoming more and more global, more and more connected. And I think that the consortia are going to start to combine more in the future in terms of all the information that's pulled in, but also the different associations are connecting on things. I know that ALS Canada is actually fortunate to collaborate with ALS Association and M&D Association on a platform that's actually a really unique one to design to try to make tools in the laboratory that typically don't work that well, really work fantastically and save a lot of time and money on the other end for researchers. But that's just one small piece. There are, you know, many other collaborations, and I think we continue to look for where we can find synergies to leverage each other's dollars to do the same things better. Does anyone want to add to that?
I just reiterate Project Mind, which was mentioned earlier.
Yeah.
Again, we've all put in a little bit of funding to make that happen together. There's many examples of that with us all working together towards the same goal. I'm sure there are more from the panel.
And I think the platform trials have also given an opportunity for countries to work together. I know there's Australian drugs in the Healy trial. I know there's an Australian arm to the NCALs. So I think those opportunities, especially perhaps for we might call smaller drugs, earlier stage drugs, are getting better opportunities now, which perhaps in some countries they might not have that. So that is providing a fantastic platform.
Great. Actually, a great one, Nick, to mention the Project Mind, because that goes well beyond the 4 societies that are represented on this particular call, and you know, so many countries, societies have contributed so significantly to Project Mind beyond all of ours. So that's a wonderful example of how everyone can come together. Jan, any other questions, or would you like us to wrap up?
You know, just Tammy asked if everybody could tell who they are if they put out a question. So we have a new question here from Karen Meistrik. Why do you think more recent research isn't targeting slow progressors? Seems like that would be very beneficial.
Well, certainly understanding why people progress slowly is something that a lot of researchers are looking into. Is it a genetic change? Is it a genetic cause that can keep them as a slower progressor? Are there certain aspects about them? Studying them in that way would be really important. In terms of clinical trials, it's more difficult because you have to be able to see a change to see if a particular therapeutic works. But I would pass this off to anyone on the council if they'd like to specifically answer that one. Anyone?
Well, I can talk again.
Sure.
So I think, David, you summarized it in a really nice way. Research-wise, preclinically, I mean, if we're not talking about the clinical trials, slow progressors are highly, highly interesting. Of course, is as you said, is it a specific gene? Are there many genes? Are there genes causing the disease and halting the progression? Is this depending on your age, your gender? Can we connect this to exposure in the environment? Or so I mean, there are extremely interesting questions that we need to figure out and work and create targets for clinical trials. But then when we're talking about clinical trials, it's a really tricky situation. Often people with, not always, but often people with slower progression might have lived for a couple of years more than most pharmaceutical companies want when they're introducing the substances. So they've already lived beyond the effect of the drugs that we are testing. And if we need to see this change in outcome, or this change in the ALS-FRS, this activity daily living score. So that means that the ones with the slow progressor, the slow progressions, that they are not meeting the outcome measures that we can target in a specific time. Maybe if we can use prediction models and we can use the patient's own predictive slope and the change in time, or the change in that specific patient's slope, we will be able to include slower progressors as well, because we can individually see the change in the future. So that would be very nice.
Okay, we're getting to the end of the session, and I see there are 2 more questions. And very well done, Alper from Turkey. Any advice on how PALS and CALS for COVID-19?
With regard to vaccination, I think he said, well, I am not an expert on COVID-19 aside from what, health officials would say. We have one physician on the panel, so I'm sorry, Caroline. I mean, if that the question would probably have to go to you.
Well, so it's very interesting. When I've been on different calls during these last couple of months, it's been very evident that there are not that many patients with ALS that are getting COVID. And if they are getting COVID, they actually, many of them will have, just a normal pneumonia as a healthy person, and the COVID-19 infection actually is not affecting the ALS. So I just want to say that firstly, because that's extremely interesting and very, very important. And in the beginning of the pandemic, I was very stressed that my patients would be very, very ill from this COVID-19 infection, but it doesn't seem to be the case actually. So that's my first. So I'm recommending all my patients in Sweden to take the vaccinations that are offered right now, the vaccinations against other influenza viruses and other pneumonias. So regarding the COVID-19 vaccination, that's a really difficult question because we don't know if it exists yet and what the side effects will be. So I think that's a very difficult question. But yeah.
I think if I can interject there, if you don't mind, if we've got time, there is some quite good information on all of our association's sites. I know the M&D Association's got some really good information and advice about COVID-19 and motor neuron disease. And I think it's really important that we understand that people with motor neuron disease are vulnerable to COVID-19, and anything that we can do to lobby and campaign to try and make governments and aware of this is really important. So it's important that people, if a vaccine becomes available, that people with motor neuron disease are able to get it as soon as possible. There is actually a campaign starting with a hashtag Vax for Vulnerable, which I think people can follow. So I think, yeah, it's something to be very aware of, and we really need to make sure that people can get access to that, so they can continue on.
Okay, we're running out of time or overtime already. I'm sorry to interrupt, which is of course very difficult. We have such experts on the line, and I need to conclude. You can all see what Michael Robinson is writing. So that's of course a very important subject: expanded access programs on variable potential medicines. From now, I think we're getting to the end. So I would like to thank the Scientific Advisory Council not only for today, but for all the work they're doing during the year, which I can see from all the emails that go up and down is a lot of work. And it's always a very balanced opinion that you give on potential medicines, and which is worth a lot to everybody in the ALS community. So we're wrapping up for a break. I'm not sure if there will be some break slides now, but before that, I want to conclude that it was a pleasure for me to be the first, first virtual moderator, which is it's quite interesting. And after the break, so we will be back at 9:15. There will be Nick Kuldip, who will be the moderator for the program for PALS and CALS. And so that's it from my side. So thank you very much to Australia, Argentina, Italy, and the Scientific Advisory Council for your presentation. And I think from now, I leave it up to the Congress what to do.